and I'll hand it off to the first tutor.
All right, hi guys. So let me start sharing. All right, can you guys see my screen? All right, cool. So I'm just gonna be going over RNA um, and its secondary structural elements and the non-standard base pairing interactions that are common in RNA. So a big theme in this class, as you guys have probably started to catch on to already, is just how regular and consistent the DNA helix structure is. As you can imagine, this is very important in regards to DNA's function as a template, since its main job is to store genetic information and conserve its fidelity. It makes sense that its structure is so regular and consistent. But on the other hand, RNA is quite versatile in both structure and function. Um, First of all, not all RNAs code for protein, of course. Not all RNAs are messenger RNAs that serve as templates. And great examples are ribosomal RNA and tRNA. They have almost protein-like roles where tRNA, for example, is responsible for recruiting amino acids during protein synthesis. So because non-coding RNA does not serve as templates, fidelity is not really a big concern, and things like modified bases and sugars and non-standard base pairs are pretty common in RNA. And as a result, non-coding RNA can fold to form a wide variety of three-dimensional structures. And you can see that pretty well when you look at ribosomal RNA and tRNA. They have pretty complicated 3D structures. And then when you compare that to mRNA, it's just kind of linear and it doesn't really fold on itself. So the most common non-standard or non-canonical base pairing interaction is called a GU wobble base pair. And as its name states, it is a base pairing interaction with between guanine and uracil. And it's even though it's a non-standard base pair, it's pretty stable. It's more stable than other mismatched base pairs, and it actually is almost kind of just as stable as a Watson-Crick base pair. They can also interact with other molecules. For example, the amino group on guanine is a hydrogen bond donor. So presumably it can interact with other RNA or other proteins. And there's also heteroatoms like nitrogen and oxygen in guanine and uracil that can bind positively charged metal ions because of their negative electrostatic potential. So one important example that where you can see the GU base pair is in tRNA, which the GU base pair actually changes the tRNA's shape and electrostatics in a way that allows it to load the correct amino acid during protein synthesis. And the details of this aren't really that important because you didn't really go over it in lecture, but basically, if you were to substitute that GU base pair for like a GC base pair, that would change the shape and the electrostatic properties of the tRNA in such a way that that its function would be affected, and it probably wouldn't be able to load the correct amino acid. So another kind of non-standard base pair is seen in what are called Hoogsteen base pairs. Essentially here, the major groove of DNA has several atoms along it that can form hydrogen bonds with other molecules. So a single-stranded nucleic acid, whether that be DNA or RNA, can sort of wedge itself into the major groove and form hydrogen bonds with it to form a triple helix, which hopefully you can sort of see in this drawing right here. The bubbles are representing the double-stranded helix, and then this strand is that third single-stranded RNA or DNA. So that's what they mean by calling it a triple helix. There's like 3 single strands interacting with each other to form a triple helix. And then this schematic right here is just showing you the specific hydrogen bonding between the Hoogsteen base pair between a uracil and an AT base pair, where the AT is a Watson-Crick base pair and the AU is a Hoogsteen base pair. And I think this can be kind of confusing because if I didn't tell you, or if this picture didn't tell you which one is the Watson-Crick and which one is the Hoogsteen, you might be thinking, well, why can't the AU be the Watson-Crick base pair? And to answer this, you have to look at the hydrogen bonding interactions and which atoms are involved in those hydrogen bonds. Because although adenine and uracil can form a Watson-Crick base pair, the actual specific hydrogen bonds are not canonical. So like, I hope that makes sense. So like if you were to have an AU Watson-Crick base pair, the adenine, these atoms would have to form hydrogen bonds with the uracil, not these. So yeah, I think this will become more clear when you guys have more practice with this, and I have some practice questions at the end. Secondary structure of RNA. How do you go from a primary linear sequence of nucleotides to the folded form of RNA? Basically, the primary sequence of nucleotides folds into like stretches of double-stranded regions that are connected by single-stranded regions. And the end result is you have like these different kinds of secondary structural motifs or elements, or I think Professor Korean even referred to them as modules. So they all mean the same thing. And these include helices, single-stranded regions, hairpins, loops, and there are a couple others that are mentioned in the book, but I think these are the most important ones. And then the most important structural motif in RNA, at least covered in this class, is called a GNRA tetraloop. So basically these are made up of 4 nucleotides. G for guanine, N for any nucleotide, R for a purine, so basically adenine or guanine, and A for adenine. And what the specific sequence does is it creates a sharp turn in the RNA backbone, as you can see here. And you might be thinking, well, this is kind of, how is this stable? How is this looks pretty unfavorable to have such a sharp turn in the backbone. Well, this loop is stabilized by non-standard hydrogen bonding and base stacking. And the book kind of goes more into detail about this, like what specific atoms are involved in these interactions. So you can look there for more information. And the N, R, and A bases, as you can see here, are flipped out, which allows interaction with other RNA and proteins. So I think for the interest of time, I'm not going to go over the practice questions, but these slides are going to be uploaded on the Google Drive. And also I have the answers to these on the next slide. So please feel free to do those on your own time. But yeah, that is the end of my slides.
All right, next, let me share my screen. All right. Can everyone see my screen?
Yep.
All right. Perfect. All right. My name is Sam, and I'll be going over protein structure and folding. So till now, you first learned about DNA, its structure, supercoiling, and then RNA, and now onto proteins. So it can be said that the basic building blocks of proteins are amino acids. I'm sure you've all heard about them. And it's pretty fascinating to know that only 20 amino acids make up every protein we know that makes up the body. And there's a basic structure that each amino acid follows. There's the amino group, there's a carboxyl group, and then there's the R group, which distinguishes each amino acid from each other. And every amino acid follows this basic structure, except proline, which you'll see in a couple of slides, because in the case of proline, um, it doesn't have a distinguishable amino group, and its R group is a cyclic ring with the NH2 attached to it. So it's different from the rest, but the 19 other ones all follow this general form. And the R group is very important because it's the only distinguishable factor between these amino acids, and they can be split into three main groups: charged, polar uncharged, and non-polar. And uh, these the characteristics of the R group determine how the protein folds and how the protein interacts with its environment. And now that we know the basic building blocks that make these proteins, how did the proteins get formed? Well, these individual amino acids, as you see here, come together through a condensation reaction. And you'll see this as a repeating pattern. Whenever you want to bring two monomers together to build a polymer, it happens with the removal of water. And in this case, when water is removed through this condensation reaction, the bond that gets formed between these two individual amino acids is called a peptide bond. And the reverse is true as well. If you want to break these peptide bonds, you would add water through something that's called a hydrolysis reaction. And as you bring together these individual amino acids together, you build long chains. And when they reach around 50 amino acids, you get polypeptides. And you can see an example here. And if you focus on this backbone, you'll see a repeating pattern. As you see with these rectangles, it's going to be NH, the amino group, the alpha carbon that with the R group attached to it, and then the carboxyl. And then this is the peptide bond. And once again, there's the amino, alpha carbon, and the carboxyl, and so on and so forth. And just for the naming, the end of the peptide backbone that ends with the amino group is called the N terminus, and the other end that ends with the carboxyl group is called the C terminus. And as I said before, to characterize these amino acids, use the R group, which distinguishes them, and they're split into two groups. The hydrophobics, as you see in the bottom here, these side chains are non-polar, so they don't interact very well with water, and that has a lot of implications of how proteins fold in solutions that contain water. And as you see here, they usually contain a lot of carbon and hydrogen. As in the case of leucine, it's just a bunch of carbons and hydrogens. Or in the case of phenylalanine, tyrosine, and tryptophan, it's large cyclic groups containing a lot of carbons and hydrogens. And then the second group are the hydrophilics, and they contain polar groups, so they can easily hydrogen bond with water. And they're usually split into two subgroups, the charged and the uncharged. The uncharged are here. These are they have a partial dipole, but they're not charged. And then there's the charged ones, which are either going to be positive in the case of arginine, histidine, and lysine, or negative in the case of aspartic acid and glutamic acid. And in this case, they split into a fourth group, the special cases. And that's because, as I said before, proline is unique because it doesn't have an R group, as you see here. It only has three things sticking out of the alpha carbon, and there's no distinguishable amino group. Glycine has no R group; it's just the H, a hydrogen atom. And then cysteine is unique because it has this thiol group that can form thiol bridges, which I don't think the class really goes into much depth, so you don't need to worry about that. But for the most part, just know that they're split into two main groups, one of which is the hydrophobics and then the hydrophilics, and that has a lot of implications in how proteins fold. Oh, okay. So now just a quick question. This is a picture I took, a screenshot I took from a problem set from last semester, and it's a picture of a protein, it's a transmembrane protein going across a lipid bilayer. And based on what I said from the previous slide, what kind of proteins would you likely see on the surface of the protein? So around here, and why? Feel free to type in the chat or just unmute really quickly.
Wouldn't it be hydrophobic?
Yep, and why do you think that?
Because the lipid bilayer is hydrophobic on the inside, and on the outside it's hydrophilic. But these I think alpha helices are they're in the lipid bilayer. So in order for this to happen, they need to be hydrophobic on the outside.
Yep, perfect. Yeah, you're right. Exactly. So you're most likely to see lysines, isoleucines on this side, and then on the inside, you're most likely to see hydrophilic ones. Perfect. Now moving on. So we just talked about the basic simple structure of protein. So just assuming that they're amino acids bonded together with peptide bonds. And this was the primary structure that I've been talking about. But in real life, they're more complicated. So then there's a hierarchical structure with protein folding. So as you build up, you get more complex. So I talked about primary sequences. And then once you bring together these primary sequences into specific folds, you get alpha helices and beta sheets. But I think the next tutor will go into much more in depth about the structure. But for now, just know that secondary structures are alpha helices and beta sheets. And then once you go into a 3D, once you put these secondary structures into a 3D fold, you get the 3D shape, which is called the tertiary structure. And then once you combine these tertiary structures together, you get a quaternary structure. And in this case, it's a hemoglobin. If you notice, this is one subunit, the tertiary structure, but hemoglobin is a tetramer and it has 4 subunits. And then, so how do these proteins fold? And what's the driving factor behind the folding into these complex structure? Well, it's the hydrophobic effect. And if you notice that these proteins are usually found in solutions containing water, and water is a polar molecule. And as I said before, the R groups that make up the amino acids have a very important function in folding the protein. So you would want the hydrophilic polar amino acids facing the water and the hydrophobic non-polar amino acids facing away from the water. As you see in the picture, you can imagine these amino acids being the non-polar ones and these being the polar ones. And this is the driving force behind protein folding. And this leads to the formation of a hydrophobic core where all the hydrophobic non-polar amino acids are moved away from water, with the hydrophilic ones pointing out towards the solution. And this also has another benefit because if you notice, as mentioned in lecture, if there's, imagine these blue things to be water and this to be the protein. If the proteins are separated, the water is limited in its entropy because they're kind of blocking the degrees of freedom water can have. But once you bring the protein together, the water is more free to move around in solution. So there's also added benefit. While the entropy of the protein is decreasing, the entropy of water is increasing. And I put this question here, but I want the other tutors to have enough time, so you can actually the answers on the next slide, but I'll just go for it myself. So as I said before, the main driver of protein folding is the formation of the hydrophobic core. But you can also think that once the protein folds, there will be internal bonding between the residues in a folded protein. As you see here, the carboxyl group of one twist is bonded to the amino group of another twist. So why isn't this a driving force behind protein folding? Well, if you look at this picture, in the unfolded state, as you can see here, these amino acid groups are already bonding with water. So in their unfolded state, there is hydrogen bonding. And then when they fold, there's hydrogen bonding again. So because of this exchange, the net hydrogen bonding energy of the backbones does not change much as the protein folds up, because in the unfolded state, there's hydrogen bonding. So there's not much gain through hydrogen bonding. And that's why if you take into account that the hydrophobic effect is going to be the main driver behind the folding of proteins. And then additionally, this is another point that was pointed out in lecture, is that isolated peptides are not very stable on their own, because for the same reason that they can't really hide away their hydrophobic amino acids away from water. And that's why you don't usually see isolated peptides by themselves. You usually see clumps of proteins together so they can form that hydrophobic core. And then moving on, I think, yeah, the last tutor kind of pointed to what structural motifs are, and they're essentially combinations of secondary structures, alpha helices and beta sheets in the case of protein, to form basic structural called structural motifs. And as with the RNA, as we saw before, proteins too have these specific structural motifs that are built from alpha helices and beta sheets. And I'll just give you three examples that were brought up in lecture. I don't think in lecture, but they were in the textbook. The first one is a helix turn helix motif, where you see an alpha helix joined by a turn and another alpha helix. And this specific structure is used to read DNA sequences. A second motif is pretty much the same, but it's two longer alpha helices joined by a loop and another alpha helix. And a specific function that these motifs have are in holding calcium, which is a positive charge. And then these helices usually have negatively charged amino acids like aspartic and glutamic acid to hold that calcium in the loop region. And then finally, there's a Greek key motif, which is four adjacent antiparallel beta strands connected by turns. And if you notice, the first and the last strand are connected by an alpha helix. And this motif is called a beta alpha beta motif. And then there'll be much more talk about the specific structure of the beta sheets and alpha helices. But for now, that's all I had. If there are no questions.
All right, let me get this going. All right, so hi everyone. My name is Chethan. As always, feel free to just unmute yourself and ask a question, interrupt me, and that way I can hopefully answer your question as specifically as possible. So Sam gave us a good intro into hierarchical, into protein structure. And right now we're going to take a deeper look into secondary structure. So alpha helices and beta sheets. So when you see secondary structure of proteins, you should immediately think hydrogen bonding from the protein backbone. So first thing we'll talk about are the alpha helices. So one thing is that they're right-handed, and being able to tell the handedness of any DNA molecule, protein, alpha helix that the professors give you is going to be really important in this course. So for this, one way you can do it is treat this as a spiral staircase and this ribbon as the handrail. And think about which hand am I going to use to grab onto the handrail as I climb upwards. In this case, it'll be your right hand, so it's a right-handed helix. Another thing to note is the hydrogen bonding pattern, of course, between the donor with hydrogen attached to nitrogen and the carbonyl oxygen. And we'll talk about this more later, but all the hydrogen bonds are satisfied when in an alpha helix or any secondary structure, so they can exist in hydrophobic areas. These hydrogen bond donors and acceptors also lead to your overall dipole moment, which I believe was on your past problem set, maybe. But if you think back to general chem, looking at electronegativity, the nitrogen is more electronegative than the hydrogen, so you got a delta minus on the nitrogen, delta plus here, and going to the other acceptor, oxygen has a delta minus, carbon has a delta plus. So overall, they'll transfer to the other ends of the helix, so you end up with a C terminus that's negative and terminus that's positive. So that's going to be also important to remember. And of course, for every turn of the helix, you have about 3.6 residues per turn. We'll come back to that as well when we talk about helical wheels. The other secondary structure we'll talk about are beta sheets. They're made of multiple beta strands. So you can see on the right, these arrows are representing the individual strands. There are parallel and anti-parallel beta sheets. This is an example of an anti-parallel one. As you can see, the arrows are alternating directions. And so beta sheets have a right-handed twist. I could not find a good picture depicting that, but if you take your right hand and kind of splay your fingers out to the side and look down on it, the way your fingers are going, they'll represent each beta strand, and that'll tell you if it's a right-handed twist. And now let's look at the hydrogen bonding pattern on this slide. So it differs actually between antiparallel and parallel beta sheets. As you can see, just based on the geometry of how the amides and carbonyls line up with each other, antiparallel, they line up almost perfectly, so it's kind of linear, whereas parallel, they have more of a diagonal, just because that's how they line up. Also, one thing to note about beta sheets are the alpha carbons. Where are they relative to these donors and acceptors? So pretend these donors, this pair is in the plane. The alpha carbons are going to be coming out of the page and into the page. So if you look at this first alpha carbon, pretend it's coming out of the page towards you. Then when we go to the next one, this one will be pointing inwards, then out, in, etc., etc. So that'll be important when we talk about amphipathic stuff. But yeah, those are beta sheets. So let's talk about satisfying hydrogen bonding. So then talked about how the hydrophobic effect is what drives protein folding. And the hydrogen bonds play no big role, as we talked about, because if you look at the unfolded state, the hydrogen bond donors, those amides and carbonyl groups in the backbone, they can be hydrogen bonded to water. And when it's folded, like here, they'll be bonded to themselves in the protein, and that's how you get these secondary structures. So regardless of if you're in the unfolded or the folded state, those hydrogen bonds exist somewhere. So there's no net energy contribution for folding. Now, just because it doesn't contribute to folding doesn't mean they aren't important. It's important that they exist somewhere. And that is why you can have alpha helices and beta sheets, maybe, in hydrophobic areas buried in the interior of a protein. Because if it's in the inside of a protein, very hydrophobic, and those, like let's say the hydrogen bond donor was not bonded to anything, that's a really big energetic penalty you're paying, and that will be very destabilizing to the overall protein. So just remember, it's important that it exists somewhere in the protein. It's just not going to be relevant to protein folding. Any questions on that? Okay. Cool. So let's talk about amphipathic helices. Okay, still got time. So amphipathic, what does that mean? It means you have both the hydrophobic and hydrophilic parts. And when we talk about helices, we refer to those as faces. So you can see an example of this over here. The yellow amino acids are all hydrophobic, and the colored ones are some sort of hydrophilic, charged or uncharged. You can see you have a cluster on this right side. This is the hydrophobic face, and left side is the hydrophilic face. So you find these amphipathic helices usually near the surface of proteins. And why would you find them there? Because when you're at the surface of a protein, one side will be facing the inside of the protein, right? And the interior or something buried in the protein is going to be hydrophobic. So if you're facing that hydrophobic side, you need hydrophobic residues. So that'll be this right side of the helix. And then near the surface, the other side, of course, will be exposed to the aqueous environment, and therefore it'll need hydrophilic residues. Now, you will be asked in class to draw, and on exams, to draw this type of helical wheel. So how do you do that? So let's come back to our 3.6 residues per turn. One turn is 360 degrees. So if you take 360 degrees and divide it by our 3.6 residues within those 360 degrees, we'll find out we'll get approximately 100 degrees per residue. So if we're going from 1 to 2, that's 100 degrees, 2 to 3, 100 degrees, etc. So we'll go over a practice problem at the end, but that's how you'll be filling out your helical wheel on problems. There are also amphipathic beta sheets, and of course, they are also found near the surface of proteins. However, compared to like amphipathic helix, it's a little harder to tell based on the sequence, mostly because you're working with fewer residues. Why is that? Because beta sheets are made up of beta strands, the full extended protein chain, whereas in a helix, you're twisting that, so you're shortening the distance. So let's take 20 angstroms. In 20 angstroms, because it's twisted, you can fit many more residues into those 20 angstroms. But if you take a beta sheet and a beta strand, you're basically unwinding that whole thing. So you can fit a lot less residues into those 20 angstroms. So that's why it's a little harder to tell from sequence. But you could expect an I+2 pattern, because we talked about how the alpha carbons, where the R group is connected, that'll be going in, and the next one will be going out of the page, right? So you can see here, this alpha carbon, this is isoleucine. It's going in, facing the interior of the protein, so it's hydrophobic. Next alpha carbon's here, the lysine pointing outwards. So it's going to be hydrophilic, and then so on, in, out, in, out. So you could expect an I+2 pattern, I+2. However, we find that sometimes it's not the case. As you can see, this valine is pointing outward. Anyways, I don't think this was terribly important when I took the course, but it's always good to know for our future classes and your general knowledge. Any questions there before we get to questions? All right, I got about 5 minutes, cool. So first question, someone wants to unmute themselves, or if the other tutors can tell me if something happens in the chat. Which is more stable, a parallel or anti-parallel beta sheet? So you can think about the hydrogen bonding pattern we talked about. Someone unmute themselves and let me know.
Would it be anti-parallel?
Yeah, and why do you think that?
Because unlike the parallel, they aren't at strange angles, so there'd be like less strain.
Exactly. They're lined up better. They might probably a little bit closer. So that, because they're right across from each other,